Bismillahirrahmanirrahim. Today we are going to learn about the structure of skeletal muscle and actin and myosin. To know about the basic structure of skeletal muscle and actin and myosin, we are going to the first slide. In this slide, we can see the cross section of a skeletal muscle. In this structure, we can see that the whole muscular mass is surrounded by a layer that is called epimysium. The epimysium surround the whole muscular mass. If we go to the detail, the whole muscular mass consists of bundles of muscular masses. You can see over here the bundles of muscular masses, and each bundle of muscular mass is called fascicle. Each bundle of muscular mass is called fascicle. Each fascicle is surrounded by a layer that is called perimysium. Each fascicle is surrounded by a layer that is called perimysium. Now, each perimysium contains small bundles of fiber that is called muscle fiber. Small bundle of fiber that is called muscle fiber, and each muscle fiber is surrounded by a layer that is called endomysium. That is called endomysium. The muscle fiber is also called myocyte or muscular cell. In this slide, we can see that this muscle fiber is magnified. This muscle fiber is magnified, which is surrounded by endomysium. This is the endomysium which surround the muscle fiber, and each endomysium contain another layer. I am going to show you in this diagram. You can see that each endomysium contain another layer. This layer is called what? This layer is called sarcolemma. This is actually the sarcolemma of myocyte. This is actually the sarcolemma of myocyte, because the muscle fiber contain the myocytes. The muscle fiber contain the myocyte, and each myocyte is enclosed by a layer that is called sarcolemma, and later on the sarcolemma is enclosed by the endomysium. So each myocyte contain the small fibers or small fibrils that are called myofibrils. These are the myofibrils which are present inside each myocyte, Going to the next slide. In this slide, we can see that this layer, this black layer, this layer is actually the endomysium layer. This is the endomysium layer, and each endomysium layer contain the myocyte. This is the myocyte. This is the elongated muscle cell. So each elongated muscle cell contain these red fiber. You can see these are the myofibril, as I have shown you. These myofibril and this myocyte has been represented over here, that this is the myocyte, and this myocyte is enclosed by a layer that is called sarcolemma. And above each sarcolemma or above each myocyte, there is a layer which surround the cell. This layer is called endomysium. So the sarcolemma is the second layer beneath the endomysium. Now each myocyte contain the small fibers which are called myofibril. So these are the myofibril which I have shown you in this slide. These myofibril are shown over here as myofibril. Now each myocyte contains small holes or small tubules throughout the cell. I have made only one tubule, but the large number of tubules are present throughout each and every myocyte. So each myocyte contains large number of T-tubules, and T-tubules are in the form of holes which provide the extracellular fluid to each and every part of the myocyte. For example, this part of the myocyte, this is the sarcoplasm of myocyte, and this part of the sarcoplasm is in connection with the extracellular fluid which are present over here. But this part of the sarcoplasm is not, this part of the sarcoplasm is not in connection with the extracellular fluid. So this part of the sarcoplasm is provided close connection with the extracellular fluid with the help of T tubule. So the T tubule provide a close connection of the extracellular fluid to each and every part of the cell. This is one function. Another function of the T tubule is to provide the action potential to each and every part of the cell. And what is the function of the action potential to be provided to each and every part of the cell? The function of the action potential to be provided to each and every part of the cell is actually to provide to the sarcoplasmic reticulum or to the vesicles of sarcoplasmic reticulum or to the cisternae of the sarcoplasmic reticulum. This is the sarcoplasmic reticulum which goes all over parallel with the myofibril. This is the myofibril, and the sarcoplasmic reticulum is all around the myofibril, and the tip of the sarcoplasmic reticulum, which is in close connection with the T tubule, is always containing vesicle, and this vesicle is called cisternae. So each cisternae of the sarcoplasmic reticulum is very rich in calcium. This contains large amount of calcium over here and over here. So what is the function of large amount of calcium in the cisternae? So if this cell, this myocyte is provided by an action potential, this action potential will be transferred through the sarcolemma throughout the cell, and this action potential will also be transferred to this T tubule. When this action potential is transferred to this T tubule, what will happen? This action potential will also be transferred to this vesicle, to this cisternae. When this action potential is transferred to this cisternae, what will happen? This cisternae will secrete large amount of calcium. When large amount of calcium is secreted by these cisternae, what will happen? The calcium level inside this myocyte will be increased, and when the calcium level inside the myocyte is increased, what will happen? That increased calcium level will result in the contraction of this myofibril. These myofibril will be contracted by the increased amount of calcium inside this myocyte. What is the role of calcium? I will show you in the mechanism of contraction of muscle, that how the calcium is responsible for the contraction of these myofibril. Now coming to the next slide to know about the actual structure of this myofibril. This myofibril consists of heavy and light chain. The heavy chains of protein are called myosin filament. The myosin filament is represented by the blue color over here. This is the myosin filament, and these are the actin filament. These are the light protein filament or actin filament. These actin filament are attached at the base with each other with the help of a special type of protein that is called actinin. In this slide, we can see that the actinin is responsible for the attachment of actin at the base, and the base of each actin is attached with the sarcolemma with the help of a special type of protein that is called desmin. Over here, I have labeled that this min is responsible for the attachment of the base of each actin, or for the attachment of actin fiber with the sarcolemma. So this min is responsible for the attachment of base of the actin with the sarcolemma, and the attachment of the base of actin is responsible by the actin. Actin is responsible for the attachment of the base of um, actin. So this is the myosin filament, and myosin filament is attached at the base with the help of a special type of protein that is called myosin binding protein. Myosin binding protein is responsible for the anchoring of myosin filament, and the desmin and actinin is responsible for the anchoring of actin. So if we go to the detail, this myosin binding protein and the desmin and the actinin, all of these are anchored by the help of a special type of another protein that is called titin. I have not mentioned in this slide, but titin is responsible for anchoring of all these protein inside the muscular cella, inside each myocyte. Now if we go to the detail of this muscular cell or these filament, this is actually the myosin filament. This is the actually the myosin filament, and this is the actin filament. The actin filament is further divided into three parts. Our actin is made up of three types of proteins. I'm going to write the names of these protein. That actin filament. I'm going to write the names of these protein. The tropomyosin, troponin, and F-actin, or you can also call it simple actin. So the tropomyosin, troponin, and F-actin are the three types of protein which are present inside the actin fiber. This is this bead-like structure, the bead-like structure throughout this whole. You can see over here the bead-like structure. This bead-like structure. This represent the actin or F-actin. This is also called F-actin. Now the tropomyosin. Tropomyosin is the thread-like structure. This is also made by the amino acids or protein layer. So this is the peptide layer. This is the tropomyosin. The string-like structure all around the beads, all around the actin or F-actin, that is called tropomyosin. And the third one, that is called troponin, which is very important. The troponin, which is made by three domains, which are having three domains. The troponin T is one domain, the troponin C is another domain, and the troponin I is another domain. The troponin T is responsible for the attachment of this protein with the tropomyosin, with this string. The troponin I is responsible for the attachment of this troponin with the actin. The troponin I is responsible for the attachment of troponin with the actin, and the troponin C is responsible for the attachment with calcium ions. As I have told you in the previous slide that the calcium is responsible for the contraction of muscle. So when calcium bind this troponin C, the contraction take place. I will show you in the next lecture. So the troponin C is responsible for the attachment of calcium. Troponin I is responsible for attachment with the actin, and troponin T is responsible for attachment with the tropomyosin. So each actin filament is represented over here, that each actin filament overlaps the myosin filament. This blue color represents the myosin filament, which is overlapped by the actin filament. If we go to the detail of the myosin filament, what is there inside the myosin filament? The myosin filament consists of two heavy chains and two light chains. The heavy chains in this diagram has been represented by the blue color. The blue color shows the heavy chain, and the two light chain has been shown by the red and green color. These are the two light chain. So this is the tail area of the this is the tail area upward. This is the tail area of this myosin filament, and this is the head area of this myosin filament. The myosin filament, or the heavy chain of the myosin, consist of what? Two heads. The heavy chain of the myosin consists of two heads, the arm area and the tail area. The tail area, the arm area, and the two heads area. So the head area consists of, over here at the base of head, you can see that the light chain has been attached. Two light chain has been attached near the head area of the myosin filament, and near the neck area, or at the lower arm, the two light chain of the myosin are attached, which have been represented by the green color. So the area of the myosin which is not covered by the light chain, over here and over here, which is not covered by the light chain, represent the hing. This is the hing 1 and hing 2, as I have shown you that the hing 1 is represented over here below the head area, which is not covered by the light chain, and over here this area is also not covered by the light chain, and this area also represent the hing area. This is the hang 2. This is the hang 2. As we go to the detail, the head area of the myosin contain two sides, and I have mentioned over here that the ABS and ATPBS. This is actually the actin binding site of the myosin head. The actin binding site of the myosin head and the ATP binding site of the myosin head. The ATP binding site of the myosin head contain ATPase enzyme. The ATP binding site of the myosin head contain ATPase enzyme, while the uh, actin binding site of the myosin is responsible for combining with the F-actin filament of the actin. We will know about the detail of attachment of this myosin filament with the actin filament and the sliding mechanism in the next lecture. Now coming to the previous slide, in this slide we can see that the overlapping of the myosin filament and actin filament give rise to certain bands, the I band and A band, H zone, the M line and the Z line, etc. The As we see in this diagram, that when the plane polarized light is passed through the myofibril at this area, the plane polarized light is passed through this area, the diffraction of the plane polarized light will be the same as the incident angle. There will be the diffraction of the plane polarized light through this area will be the same as the incident angle. But if we pass the plane polarized light through this area where there is overlapping or where there is myosin filament, the diffraction of light will be the angle of diffraction of the light will be different. So this area where the angle of diffraction is the same for the plane polarized light is called the I band, and this area where the diffraction of the angle of the angle of diffraction of the light will be the same for a plane polarized light or different for the plane polarized light is called A band. The angle of diffraction will be different in this A band, and the angle of diffraction will be same in this I band. That is why this is called I band, because this represents the isotropic area, and this is called the A band because this represents the anisotropic area due to different diffraction angle. Now the isotropic area also contain a line that is called Z line. Over here a line is there that is called Z line. The Z line is actually the line at the attachment point of actin, and Z line was here over here at the attachment point of actin. So the area between two Z line is called sarcomere. This is the basic unit of muscle cell, the sarcomere. Sarcomere is the area between two Z line, and Z line is the line at the attachment site of each actin fiber. So the M line is the attachment site of what? The M line is the attachment site of the myosin filament. The attachment site of myosin filament gave the M line. Now the H zone. The H zone is the zone. This is the H zone. This area shows the H zone in which the at the mid the M line is there. H zone is the zone inside the A band where the light can pass, where the light can easily be passed, and which is not so thick, which is not which is not showing any overlapping, and where the cross bridges are also absent. The H zone is the zone which is given or which is shown by the myosin filament inside the A band, where there is no cross bridges, and where the passage of light is comparatively high as compared to this area. And in between the H zone there is a line that is called what? That is called M line. The line which is called M line.